practical histology lab in urinary system. In section in the kidney, we can see different regions. First of all, the outer surface is covered by a connective tissue capsule called renal capsule. And we can differentiate two portions. The outer portion beneath the capsule, characterized by the presence of rounded structure, this is called the cortex, due to the presence of renal corpuscles. The inner part, which devoid of rounded structure, only contain long tubular structure, is called the medulla. So we have the outer surface, so the previous section stained by H&E stain. This is stained by a pass stain. The pass stain identify the outer membranes of the tubular and the glomerular structure. So we have outer surface and the cortex due to the presence of round structures of the glomeruli, and here we have the medulla outer region, and we have the inner region of the medulla, which is called the medullary pyramids, that contain a tip called renal papilla or area cerebrosa that project to do minor calyx of pulvicalcial system. Minor calyx is lined by transitional epithelium. In higher magnification, within section in the cortex, we can see the round structure of renal corpuscle containing a centrally glomerulus, tuft of a glomerulus, that's surrounded by a capsule called Bowman capsule. The outer part of the capsule, called the parietal layer of the capsule, that lined by simple squamous epithelium. The inner visceral layer of the Bowman capsule is intermingled with the endothelial cells of the glomerular tuft. Here the space called urinary space, capsular space, or Bowman space. The remaining part of the cortex containing tubular structures, these are sections when the tubules lined by acidophilic cells and containing a brush border. This is a section in the proximal convoluted tubule. So these are proximal convoluted tubule. Other type of tubules in which the cells are less acidophilic, more basophilic, and they lack the brush border, these are called the distal convolute tubule. Here a distal convolute tubule, this is another distal. So we can differentiate between the proximal, which is acidophilic with the brush border cells, while the distal is basophilic cells, less in height and lack the brush border. So we have glomerular capsule, a capillary, and the podocyte, which is the visceral layer, cannot be differentiated. These are proximal convoluted tubules, and this is a capillary. You can see this is found within the cortex. We have different vascular structure within the cortex. If you can call the intralobular arteries, that gives rise to the efferent arterioles. Okay, the blood-urine barrier, it is really at the ultrastructural level. Here we have the lumen of the capillary of the glomerular tuft, which is a fenestrated capillary. You can see the endothelial containing spaces between them. So this is a fenestrated capillary with diaphragm really. And we have from the outer surface, we have the pedicles or secondary processes of the podocytes, and this is the primary process. So we have the endothelial cells lining the capillary, and we have the pedicles, photoprocesses of podocyte, and their fused basal lamina. This is the filtration barrier. And in addition, the true filtration barrier was considered to be a slit membrane that bridged the space between adjacent pedicles. So we have in between adjacent pedicles, we have a slit membrane, which is a true selective barrier. This is a section in electron microscope, transmission electron microscope. So we have the outer layer of the glomerulus that's made by simple squamous epithelium with the nuclei are flattened. Also within the cortex, we can see extension from the medullary region in form of extension of collecting tubules. This is called medullary rays. These are found in the cortex in between the glomeruli, and the convolute tubules. In higher magnification, we can differentiate in the cortex presence of renal corpuscles and part of the collecting tubules. This is a collecting tubule part extend toward the cortex, which is called medullary rays, while this is a convoluted tubule, proximal convoluted tubule. In this figure, we can see the renal corpuscle, and this is the efferent arteriole of the renal corpuscle. Also, we can differentiate proximal convoluted tubule and this region of distal tubule, elongated section. This is a distal convoluted tubule, like brush border, while this is a proximal and this is a distal section of the distal that adjacent to the vascular pole of renal corpuscle, in which the nuclei found in single row called the macula densa. So the renal corpuscle containing a vascular pole that we have adjacent distal tubule in which containing a row of nuclei of macula densa. This is another section showing the renal corpuscle, and this is the efferent arteriole, and this section is part of the distal tubule that's showing the macula densa, and this section is part of the proximal convolute tubule. You can see the acidophilic cytoplasm of the lining epithelium and the brush border. Sections in the medullary region, the upper part of the medulla showing this structure, which is a part in the thick section of loop of Henle. You can see most of them lined by cuboidal epithelium. In between, you can see capillaries, which is part of the vascular structure of the medullary interstitium. In longitudinal section, we can see a duct that with wider lumen and lined by low cuboidal epithelium. This section in the collecting duct that found within the within the medullary region. So this is a longitudinal in the section in the medulla. We can see thick segments, and within it we have a collecting duct. In the lower part of the medulla, in addition to thick segments that lined by cuboidal epithelium, we can see thin segments. These thin segments are prominent in the juxtamedullary nephron, which is important for increasing the interstitium osmolarity. In between, we have capillaries, which is vasa recta capillaries, which is found adjacent to thin segment of loop of Henle. So in lower medulla, we have thick segment and thin segment, and capillaries part of vasa recta. This is section in a collecting tubule, not in thin segment. This large diameter, wide lumen lined by cuboidal epithelium is a collecting tubule. The ureter, which is lined by transitional epithelium, have a lamina propria and containing inner longitudinal and outer circular muscle, and it is covered by adventitia. The urinary bladder also lined by transitional epithelium, which is characteristic of the urinary system, and have a lamina propria and type of smooth muscle called detrusor muscles. The epithelium, transitional epithelium, characterized by presence of dumb cells, which is characterized by binucleated cells that's found in the luminal surface, have a capability for extension and reduction in size. This epithelium starts from the pelvic calyceal system to the ureter to the urinary bladder and also extend toward the urethra. Thank you.